start now, so whenever you're ready.
All right. So, good evening, everyone. I'm Alex. I'm going to be presenting my honors project using seabird wing strokes to identify important marine conservation areas. So, for a bit of context, the Canadian government committed to protecting 30% of our oceans by 2030, an ambitious goal, and for that to see which places to conserve, identify the areas of high ecological importance, I suggest a turn to seabirds. So why seabirds? Well, their natural history is linked to fish stocks. They're linked to the health of the oceans. The health of the population will be dependent on the health of the oceans in which they forage. They are conspicuous and span the entire globe, so they are easy to study, and something that is relevant somewhere on Earth can be applied somewhere else because oceans are everywhere and seabirds are everywhere. And so the link between their natural history and fish stocks or the ocean's health is their foraging behavior. So we can study their foraging behavior to gain insight on the health of our oceans. But foraging behavior in wild animals is notoriously hard to observe, so that's why we have biologgers. Biologgers are little devices that collect enormous data sets, in this case on foraging behavior. So such biologgers can be found as beak opening sensors to detect prey capture attempts, or esophageal temperature readers that detect cold fish as they enter the esophagus of a seabird as it is ingesting it. In our case, we're looking at GPS accelerometers, which measure acceleration, body acceleration, and global positioning. So we know how the bird is moving and its whereabouts, and with minimal invasiveness. So why do we want to care about wing stroke frequency? Well, body acceleration will give us wing stroke frequency, the frequency at which they beat their wings, whether that is fast or slow, so wing beats per second. And we can calculate that from acceleration data, but also we know from the literature that wing stroke frequency varies according to body mass. So an increased body mass should increase wing stroke frequency and vice versa. Now we'll go into the physics of this just in a moment, but for now, we know that changes in wing stroke frequency throughout a foraging trip can give us insight on foraging success, because we can know when the wing stroke frequency varied and therefore the body mass, and we can know temporally when prey was captured based on the gain or loss of body mass. But now with GPS accelerometers, with GPS data, we can know when, but also where prey was captured based on the variation of wing stroke frequency throughout the foraging trip. So we all remember force equals mass times acceleration. In this case, in a bird flying, you have a downward force that is guided by body mass and gravity. So that is our downward force, and we need a counter force, which is lift, which is provided by the wings. Every wing stroke provides lift by pushing air down, and every wing stroke provides the same amount of force. So that's important to retain. So if body mass increases, so must lift. And because every wing stroke is the same amount of lift, they need more wing strokes per second to keep up their flight with additional weight. So here are the objectives. We want to first validate the method of determining mass based on wing stroke frequency. So we want to know that indeed we can read mass into accelerometer data. Second, we want to apply the method to wild foraging birds to map areas of high ecological value. And so if wing stroke frequency accurately indicates body mass, then wing stroke frequency can be used to map these areas. So a bit background on the study species. The black-legged kittiwake is a small species of long-lived gulls. They exhibit biparental care, where males and females incubate the eggs and feed the chicks, and they are cliff nesting seabirds that live in colonies, found in Arctic and subarctic regions. Now their breeding season starts in April and spans May, for the pre-laying part of the breeding season, where they build their nest out of plants and mud, and then they incubate around June, and then chicks hatch in July, and the fledging occurs throughout the month of August. Now for the study site, Middelton Island is a tiny 8-kilometer long island on the edge of the continental shelf in Alaska, situated at the 59th latitude, has mostly maritime tundra biome, and is a tiny island, really pretty place. And the study system is a tower decommissioned from the Cold War era, from the 1960s, and the top layer has been upfitted to accommodate cliff nesting seabirds on the outside walls. And from the inside of the tower, we have access to each of the nests via one-directional windows. And so there's 500 windows, one pair per window, a lot of birds to which we have access to study with minimal disturbance. And 72 of the windows, 72 of the pairs, are fed, part of a food supplementation program that's been implemented since 1996. So the food supplementation consists of feeding these birds 3 times a day as much fish as they want, either capelin or herring, depending on the season, for breakfast, lunch, and dinner, basically. And that feeding will be instrumental in the validation of the wing stroke frequency method as a proxy for body mass. And I'll explain why in just a moment. So for the deployment, I deployed GPS accelerometers that we can see on the tail feathers of the bird here and there. They've been deployed for 3 to 4 days, sometimes longer when we weren't successful at retrieving them right away. The GPS accelerometers recorded acceleration 50 times a second, so that's enormous amounts of data, and GPS coordinates once every 3 minutes. So every 2 minutes, it pinned the location of the bird and registered that information in the device. So to validate the wing stroke frequency method with the fed birds, we use 66 fed birds to which we know their feeding data. So we know how many fish they ate and how much weight does each fish weigh, so how much mass each fish has. And the fish have been standardized to reduce variability in the mass of each fish that were fed to the birds. So we know closer to reality what is their stomach contents. And we measured the mass of each bird on deployment and retrieval to have a concrete measurement of their body mass. And this is like a diagram which people refer to when feeding birds that were out on deployment. So if you were feeding a bird that had a GPS on them, you made sure to feed standardized sized fish. And then on retrieval, we took off the GPS, took morphometric measurements, and then took wing photos to control for wing surface area, which controls how much lift they get from each wing beat. So that will affect the frequency at which they beat their wings, because it's the lift provided by the surface area that will affect that. And so in terms of sample size, a total of 140 birds were sampled throughout the season, and we tried to keep it even between males and females and fed and unfed birds. We have way less in incubation because we got hit with a wave of botulism, so we had to stop midway and then keep going later on, but we still got a pretty decent sample size. So this is preliminary results. This is the track of 18 birds that I selected to show the span and scope of the foraging areas that they've been visiting. So they can go really far off the continental shelf and go sometimes really far inland. Each point is a GPS sample, and the colors represent travel speed. So where red is super slow and green is fast. So we can see where they've been staying and where they've been going. So here is for the validation of the wing stroke frequency method. It should follow this proportionality. So the frequency ratio should follow the square root of the mass ratio. So let's say we have a bird that weighs 420 grams and has a wing stroke frequency of 4.25 hertz, which is arbitrary numbers, but close to reality. And that bird was fed 3 fish that we know the mass of. Then we should expect the wing stroke frequency to vary this much. So we have all this data. We have the data we have the wing stroke frequencies, we can measure them, and this is food supplemented. So we know the mass of the fish that they ingested. Now, if we don't have the mass of the fish, if we're looking to actually find that value in wild foraging seabirds, then we can invert that formula and isolate for M2, find the mass based on the wing stroke frequency. So here is another foraging map, and we can see some clustering here, here, there, and here, where clearly the birds are moving based on the speed of their travel, but they're moving kind of like irregular patterns, which suggest that they might just be foraging at those locations. And so if we have a wing stroke frequency measurement at the beginning and a body mass, and then have another wing stroke frequency, then we can plug in these values into this rearrange equation, and then get the resulting mass from their wing stroke frequency before the foraging event and after the foraging event, thus giving us the amount of fish that was ingested throughout this foraging event. And so by doing this, we can know how much fish was eaten, or how much weight of fish was eaten, and where it was captured in the foraging grounds of these seabirds. So I would like to finish by coming back to the title of my presentation, using seabirds' wing strokes to identify important marine conservation areas. By knowing where these birds are being successful at finding forage fish, we know which areas to protect. That's it. Thank you.